I am also doing research into adoptive T-cell therapy and specifically CAR therapy as a treatment for acute lymphoblastic leukemia. And my PhD project specifically goes a bit more basic because, of course, it has shown unprecedented responses in clinical trials. I think the building blocks of these chimeric antigen receptors that we use are still not fully understood or are still very much a black box. And to make this therapy more broadly applicable to other tumors, I think it's very important to understand all these different building blocks and how they behave and how they affect the T cells that should then kill the cancer cells. So I particularly focus on the binding domain, so the domain that actually recognizes my tumor cell, and how the kinetic properties of that binding domain affect the T cell's behavior following binding. So we know that there are several scfvs available against CD19, which in our case is the target for leukemia. And several have been used in clinical trials and taken forward, but no one has actually compared the actual effect specifically of these different scfvs. And although it's been very successful, I do seem to find differences in their behavior, which might not be relevant maybe within the leukemia field, as it has been so successful. But as I said, to take this forward might be of greater relevance targeting other tumors with lower levels of antigen expression. CAT's been developed by UCL within Martin Pule's lab, and initially, I think we tried to just show equivocal efficacy, but then ran into differences that we thought were because of the different kinetic properties of CAT. CAT, when it binds very strongly to the tumor cells, but also releases really quickly. And if we think there's an advantage there, you can imagine if a CAR T cell sticks to its target for too long, it might not be able to then target the next cell and the next cell and the next cell. And we have data that suggests indeed that this CAT CAR T cell is able to kill or have more interactions with tumor targets and therefore have a greater killing capacity, which we think might be an advantage. Yeah, so we first tried to make this story as complete as possible. There are still questions to be answered. And it's definitely an area of interest for me to build on my future scientific career. It's been really exciting working in this great density of expertise. I felt very privileged I'm Dutch, working at UCL. I felt very privileged to be surrounded by such great expertise, and hopefully the findings coming from the States to UK and then hopefully will follow into Europe. So it's great.